So this will be a substantially different topic. We've heard a lot about, in the last presentation, about things that we can be doing right now to change the course of the epidemic. And what I'm going to talk to you about now is an approach which we really could use to help end the epidemic. And that's the strategy to generate a vaccine. I think both are important. We have to be working diligently on each one. But I want to tell you about why my colleagues and other scientists, many of whom are here at the meeting, are more optimistic than we have been in years that we can really build a vaccine for that objective, which is to end the epidemic. So I come to you from the International AIDS Vaccine Initiative. It's a group, as the name suggests, which is focused solely on HIV vaccines. It's international. We have sites around the world. I'll mention that more at the end of the presentation. We're only a part of the effort. There are many other groups that are involved in HIV vaccines, and I'll mention how they're coming together and collaborating to try to achieve this very difficult but important objective. So why are we interested in vaccines? And the reason is that they have been probably the most successful public health intervention ever invented. They've been responsible, as we all know, for the eradication of one of the major killers throughout history, smallpox. And where they've been effectively deployed and a vaccine has been available, they've really been remarkable at reducing the numbers of the incidence of the disease. And here's just three examples from this is from the data available from the United States, where we went from a situation, where we had a substantial number of people severely infected with polio to the case where we have none. And that's completely due to the fact that a vaccine became available and was deployed throughout the country. Similarly, with measles, there's a very prevalent disease of children and impacted both morbidity but also mortality. And the incidence of that in this country has declined really substantially. There's only 44 cases reported last year in the US. Similarly, diphtheria, a deadly disease, has also been substantially reduced here, and there was only one case of diphtheria in the US last year. Wouldn't that be a wonderful situation to be able to report about HIV in the future? And that's what a vaccine is. Those of us who are working on a vaccine, that's our mission, that's what our dream and our goal actually is. Here's a model for the impact of a vaccine on the epidemic. And it's an estimation, of course. If we were to introduce what happens to the epidemic, and this is these are global numbers, about if we continue on with the existing programs that are in place, and there are no unexpected events that occur, like major resistant strains of HIV occur or something along those lines. But with continued progress, we'll see a leveling off of the number of HIV cases around the world. What will happen, and that there's no reason to expect that will change very much unless something radically different changes in the population. And there are approaches to doing that, as we've heard in the last presentation as well. If we were to introduce a vaccine, even one that had relatively low effectivity, so this is a vaccine that would only be 50% effective at preventing HIV and would have only modest coverage, 30% coverage, we'd have a substantial reduction. About a half of the number of cases of HIV would be eliminated by a vaccine with sort of modest levels of performance. And of course, a much better vaccine would cause a dramatic change in the epidemic. What we're hoping for, of course, is to combine in the future a vaccine with progress in things like PrEP and treatment as prevention, other kinds of preventive measures, better education, and together we think there's a reasonable chance that the epidemic could be diminished in a very dramatic fashion. So that's our goal, that's our dream, And how are we getting there? What's the promise of actually getting there? I'll get to that in just a second, but I want to remind you of the challenges facing HIV vaccine design. Oh, I should mention that if you're interested in these kinds of models, you can go to the Global Village booth that's there for AIVI, and there's some information on that. So the challenges that are facing HIV vaccine design are threefold. And one is that there is an incredible diversity in this disease around the world. We think of it as one disease, but it's actually many, many different viruses that are being transmitted. So this is a sort of pictorial representation of that. This is the genetic differences in the virus, and you could measure the amount of difference is reflected by so this would be one isolate of HIV on here, another isolate over here, and the extent of the difference is measured by the distance along the lines. So you can see that there are, for the isolates of HIV from this one study, you could trace long distances on this kind of spider diagram here. In contrast, a disease that we're very familiar with, influenza A, in a similar time period, had a dramatically different amount of difference between the isolates. So in a sense, we're facing a disease which has learned how to become very diverse, how to change rapidly, and how to avoid both the immune system of people, and unfortunately, so far, it's avoided our ability to generate a good HIV vaccine. But we're learning how to overcome that challenge, and I'll mention that more. The other fact is that even inside a person, the HIV virus changes very rapidly. It moves from one kind of virus and then it can actually change and avoid the immune system. So that within an HIV-infected individual, there could be thousands of different viruses circulating. And this again presents a challenge both for transmission, because there are many different viruses that could be transmitted, and it's also a challenge for control. The immune system has a hard time controlling HIV because it's constantly changing. And then third, as we all know, this virus actually infects and can destroy the very cells that are designed or that are present in the body to control infection. The T cells, the CD4 positive T cells, are part of our immune system. And since HIV gets into those cells and can disrupt their function, it's actually preventing the body from rejecting it. So we have a lot of challenges in terms of designing a vaccine appropriately, but each one of them we're beginning to see the light and see some hope, and I want to tell you a little bit about that. So how might an HIV vaccine work? This is how we're beginning to think. And well, we go to the biology of that virus, and you've seen pictures like this before, but HIV is kind of a ball that has little lollipop structures called envelope that stick out from the surface. And those proteins are responsible for binding to the surface of cells, to CD4 receptor, and another one called CCR5. And then the virus actually enters into the cell, and it uses the cell's machinery to replicate itself. And then the virus buds out of the cell and continues on to infect other cells. So what we're trying to do with a vaccine is figure out how to block each step as best we can in preventing this chain from occurring. I'll just mention briefly that antiretroviral therapies primarily work by preventing once a cell has become infected, they block the replication and the generation of the next generation of viral particles. So they can't actually they can't actually prevent this entry process. They can make the next generation much less efficient or block it almost entirely. So when we're making a vaccine, we're using two arms of the immune system to prevent infection into cells. So these are, if we could block the virus from entering through the CD4 receptor, we could obviously, hopefully, prevent infection or make it much less likely. And this is a process where we're using antibody responses. You know, the immune system generates proteins that called antibodies that block the function of pathogens. Well, we're designing antibodies and have a lot more information about how to do that now, and I'll mention that. To prevent this step in the process of how the virus gets into the cell. The other step in the process where we're making real strides is to be able to actually kill these cells that are infected by the HIV virus. And then, obviously, by killing those cells, we prevent the next generation of virus from being formed. And this is a process called cell-mediated immunity. The body has a process to do that. It's how it clears disease naturally.
Can you repeat that?
Cell-mediated immunity. And this is part of the natural process by which the immune system clears disease from the body. And we can design vaccines that actually work to improve that and have that process, to have that part of the immune system in place so that at the first stages of an infection, it could overcome the first rounds of replication of HIV in the body. So we're using both arms of the immune system, and this is a strategy which investigators around the world are exploiting. They're trying to harness both arms, and I'll tell you a little bit of information about why we think we're making progress. So on the antibody media side of things, so you remember those lollipop structures on the surface of HIV that are critical for its functioning. We have information now about how antibodies can bind to the surface of that envelope protein and block its function. And these are called broadly neutralizing antibodies. You'll hear discussions about that in other sessions. We now know that people can make antibodies that block the whole range, or the great extent of the range of the genetic diversity, the diversity of HIV that I mentioned in one of my first slides. So there are single antibodies that can block many different strains of virus. And we can locate the site of where they bind to the envelope protein. We're grouping them into a number of different sites. And these are things that are too small for you to read, but I just wanted to give you a sense that we are understanding the exact molecular structure of how that antibody binds to this envelope, this critical envelope protein. We have ones here and here and here and here and here, and that's giving us a clue as to how to build a vaccine. Because, of course, if we can now take those sites that are bound by the broadly neutralizing antibody and turn that into a vaccine, give it back to a person as a vaccine, the immune system we hope, will form those kinds of antibodies and generate a vaccine that will block infection. So there's a lot of enthusiasm right now, and I think you'll hear more about it at the meeting, and certainly there's an upcoming meeting in Boston of AIDS vaccine researchers from around the world, and this will be a very hot topic, and you'll be able to hear more about that over the next couple of years. Another area that's very hopeful is, I'm sure you all know that there was a vaccine trial that was run in Thailand that showed partial efficacy, about 30% decrease in the rate of acquisition of infection in people who received a vaccine compared to the control group. And those results were reported a couple of years ago now, but just recently an important publication occurred in the New England Journal on that same clinical trial, but exploring what types of immune response occurred in the people who received the vaccine and were correlated with, or we hope were responsible for blocking infection. And this is a copy of the paper, and you can go online and find it if you want to read it in detail. But again, what was found was that there seemed to be an association between a particular type of antibody and the ability to prevent infection. Now, this kind of information is And there's a lot of work to substantiate that and follow up on it. But this kind of work is very important because as we design vaccines and we test them both in preclinical models, animal models, or in people, we really want to know whether the vaccine is working or generating, effective responses, effective protective responses before we test them further. It's expensive and difficult to do a major clinical trial in thousands or tens of thousands of people to see if you block transmission. You'd like to know whether you can block it, whether you're generating the kinds of immune responses that you want, and this is giving us the first clues of the kinds of immune responses that we think we want in a vaccine. Another thing that is, I think, very encouraging that we can look forward to in the next few years is that we'll continue to get data from clinical trials conducted in people in various places around the world of different types of vaccines. So we'll learn more about the kind of, again, the activity of the vaccine and be able to understand what kind of vaccine we want to build in our next generation. So the next clinical trial that's likely to give us results is one called HVTN-505. It was started several years ago, and we're expecting to hear results from that sometime late next year, possibly early the following year. This is a major study that's being conducted in MSMs in the United States, and it's being very carefully controlled and conducted so that we'll get the maximum amount of information about how the vaccine is working and hopefully be able to learn from that in the building of the next generation of vaccine. There's going to be a repeat or a efficacy trial planned of a vaccine that's very similar to the vaccine that was tested in Thailand, so-called RV144 trial. And this is a group of collaborators called P5, which will repeat that study probably in sub-Saharan Africa with a vaccine that's been designed specifically for the epidemic in those regions. There are many early-stage clinical trials. As you all know, that when you have a new therapy or vaccine, you have to start with very careful testing of the safety of that product and then move gradually to testing it in thousands of people. It's not appropriate to take something into many people unless you know that it's safe and it's actually performing the way that you think you planned it for it to perform. So it takes a long time to get through those stages, and this last stage is usually the efficacy testing stage, where we understand whether or not the vaccine is actually preventing disease. So there are many new strategies that are working their way through that process in the early phase, in the safety testing phase, and the next steps will be to expand that safety testing beyond and a few people, tens of people, into hundreds of people. So that process is occurring. We can look forward to a lot of progress in the next few years in that setting. And then finally, there are animal models of HIV, primarily in non-human primates, where there's a very similar disease. It's called SIV. And in that setting, we have vaccines and have tested vaccines that are quite effective, both at preventing infection and in controlling infection. I mentioned those kind of two arms of the immune system, one blocking infection, one controlling infection. And we have strategies that are quite effective in the non-human primate. And many researchers are trying to pick the best of those to take and make a human vaccine, an analogous human vaccine, and begin the testing program. Yes. So there's a lot of progress. There's a lot of things that will be coming along in the next few years. There's renewed optimism, and I look forward to that time. I want to just mention one thing about another more non-scientific thing that has happened in the field, which I think is very beneficial, and that is that groups that are working on HIV vaccines have really become very partnership-oriented. This is a map of the world showing the sites where AIVI is actually functioning, and that's the organization I'm with, and I'll mention other groups as well. But just as an example, it's really critical to study, if you're building an HIV or trying to build an HIV vaccine, to have sites where the epidemic is most prevalent and biggest problem. And AIVI has clinical trial sites in Africa. They were absolutely critical in the very most basic discoveries that were made of those broadly neutralizing antibodies that I mentioned, because it was sera from people, blood samples from people who were infected with HIV, where we discovered the very broadly cross-reactive antibodies that I mentioned. Those antibodies actually required sophisticated technology at biotech firms on the west coast of the United States in a laboratory. The samples were transmitted through a lab in London and categorized and characterized, and discoveries were made in a laboratory in Brooklyn, New York. This is characteristic now, I think, of the kind of thing that you'll see in HIV vaccine research over the next few years. It's a global effort. It's a group. It's the alliances from around the world, and we're extending that. Now we're going to be starting a laboratory in Delhi, India, that will expand our reach to both explore that epidemic, but also bring to this challenge, the very talented group of scientists who are in India. I just want to mention several other groups that have these kinds of global networks. The National Institutes of Health of the U.S. has just announced 2 major new grants. They're called AIVI grants. That's Centers for HIV, AIDS, Vaccine, Immunology, and Immunogen Design. So it's a big acronym. The 2 very large grants have just been awarded, and they will be focused on the building of new vaccines, and they're all collaborative projects involving researchers from around the world, and that's a great thing. Another one is the Campaign for AIDS Vaccine Discovery, which is an arm of the Bill and Melinda Gates Foundation, and again, they have a network of investigators around the world. So this idea of collaboration and global networks is critical to, I think, the next steps that we need to take. So where are we going? I always get the question of how long will it take? It will take a long time. We all have to be patient. I think if we had the vaccine right now, if it's one of the ones that are in the clinic right now, it will take another 10 years before we can see it deployed in the field. That's an unfortunate and difficult time frame to be looking at, but it's one that I think we have to do if we're going to see a vaccine. Any vaccine would take that long. It's not just HIV. Persistence, with a time frame of that length, we just have to be persistent. And as we make scientific advances, we are going to have to make sure that we have the resources, and both in terms of money, but also in terms of scientists, young scientists who are willing to take this problem on. We have to build a pipeline. And the reason for that is that one thing that has plagued HIV vaccine discovery and development has been that the field tends to shift from one approach to another. And what all the clinical trials have taught us is that we learn from each one that goes into people and is tested. And I think that the best way to handle that, especially given the long timeline that's necessitated by clinical trial planning, is to have multiple projects moving through. So have different types of science that are supporting, that are at the basis of each vaccine to move forward. So we need to have multiple vaccine attempts moving at the same time. And that's good, that's necessary, and I think what is going on, but it has to be sustained. And then I mentioned partnership. It's crucial to bring all the different types of expertise and funding sources together to stick with it to achieve the goal that we're really looking for. And with that, I'll finish and be happy to answer questions.
Mr. King, thank you very much. I'm here. Just one question before we go to the next panelists, and then we all can gather together in the restaurant and talk among ourselves and with the experts. When the researchers are working on the trials, what are the factors that determine where the trial is going to take place, the sample of the population, how many people, how many are going to be males or females, and what age sample are going to test? What's the process? Because we need to understand the process you followed to get to that point.
Yeah. So it depends on what the purpose of the trial is. So as I mentioned, the first steps that are taken for a clinical trial are always focused on safety. So the first question we want to ask is the vaccine safe? And mostly that can be conducted pretty much anywhere. It doesn't really have to be done in any particular population. We think that a vaccine that is safe in one population is very likely to be safe in another population. That parameter would need to be confirmed, but I think that mostly that it's considered that we can do trials pretty much anywhere. In terms of the population that is in the trial, usually we look for a balance, so we don't have a big difference between males and females, or one age group or another. The most important thing is to have people have informed consent. They need to understand that they're part of an experimental study and are participating in something that we don't know whether it'll work. In some cases, we don't even know that it will be safe. They'll get good medical care during that process. So the first phase is really focused on just generating that kind of information. The next stage, as you go into a larger study where you're really trying to understand what's happening to the immune system, in that case, you really want to be in the population, as much as possible, you want to be in the population where the vaccine would eventually be deployed. So we think that it's appropriate to be doing clinical trials in populations in Africa. And in fact, I think been very successful. Our sites are excellent, and we think that that's the right approach. And then finally, when you're doing efficacy testing, you have to be testing in a population where the incidence of the disease is high enough to be able to measure infections. And that might seem easy, but if you were testing on the wrong part of Washington, D.C., for example, you could test forever and really never see any infections, or test for a very long time before you ever see infections. So you need a relatively high incidence population.